The California Department of Food and Agriculture is all about protecting farms and farmers and promoting the healthy, nutritious harvest they provide. To do that, we have to know what the risks are, what might come along and eat, infest, or otherwise damage crops before they make it to harvest. The protection of agriculture begins with our ability to readily identify pests, diseases, and other potential threats. We ask several basic questions. With insects, are they pests? With plants, are they weeds? With diseases, do they threaten crops? With seeds, are they the right variety? And with nematodes, are roots at risk? That's where our Plant Pest Diagnostic Center comes in. Packages and deliveries and drop-offs roll in daily to the center. Bugs, weeds, leaves, roots, snails, seeds. They all cross the front desk at the lab and move right along to our scientists for identification and analysis. And the main question is always, does it pose a risk to our agriculture or the environment? That decision often hinges on comparing incoming samples to our collections of insects, plants, and seeds, as well as creatures like nematodes and even aquatic mussels. Collecting isn't just for the scrapbook set. Here at Cdfa, our collections are the vital first step toward identifying and ultimately responding to everything from a fruit fly infestation in the Los Angeles basin, to a plant disease and vegetable crops in the valley, to a quagga mussel clog in a local irrigation canal. To address such a wide variety of threats, Cdfa's lab boasts more than 20 PhDs in various fields, all under one roof. They come from around the world to work for Cdfa, and the breadth of their collective knowledge puts this facility near the top of a very short list, nationally and internationally.
And all these flies look very similar. They are hard to identify. The genus Spectrocera has over 600 species worldwide, mainly in Southeast Asia. And some species are very hard to identify. For example, the emerald ash borer, it's a jewel beetle which attacks ash trees, and we are very worried that it comes into California. The problem is this pest species belongs to the genus Acrylus, and there are thousands of species of Acrylus which all look very much the same all over the world. We also have to deal especially with invasive aquatic freshwater mussels. They can become a problem when you suddenly have hundreds of them breeding in your pond and eating your aquatic plants.
Our collections also encompass DNA samples of fruit flies and other pests stored at 80 below so that we can identify not only the species, but also where in the world it came from. Our entomologists, biologists, and other scientists use cutting-edge tools like this electron microscope that can be magnified thousands of times and viewed on a computer screen. Here they're zooming in on an egg mass laid by a grapevine pest called the glassy-winged sharpshooter. This centrifuge and DNA analysis equipment are the workhorses of our ongoing effort to detect and rapidly respond to a devastating citrus disease known as Wong Long Bing, which is also known as citrus greening. But a lot of the work is still done by steady hands and trained eyes. Before some samples can be prepped for automated analysis, they must first be scanned for disease symptoms, like these citrus leaves that have been submitted to our lab because they may show signs of Wong Long Bing. The samples that come in aren't just insects and plant diseases, but also things like weeds and seeds.
Okay, this is the Cdfa seed and fruit collection, and it's the second largest such collection in North America after the USDA collection. We have 50 to 60,000 specimens here. Samples come into the seed laboratory every day, both from border stations or dog team intercepts or regulatory samples that are collected by our staff being sold to farmers in California for our identification. For regulatory samples, we look for contaminants like weeds and hopefully not noxious weeds that might be in with the crop species and the purity of the seed. So these are the seeds that we actually took out of tropical fruits that were stopped coming in from the Mexican border, a species we had never seen before. Here in the germinator, we have examples of weed seeds that were found in our samples of oats in the lab. And this is a jointed goat grass, or eagle ops, a noxious weed. And you'll see that it's been growing readily right upon being planted. Here's a halogeton plant that's right out of the mailing envelope from the border station. It's common in the state of Nevada, and we want to keep it out because it's very toxic to livestock. And it looks just like the dried plant.
Cdfa's Plant Pest Diagnostic Center contains millions of specimens. But this is no library, and it's no museum either. It's a hub of nonstop activity, because identifying a pest, disease, weed, or seed today may give us the upper hand on an infestation that could be devastating to our farmers and our food supply tomorrow.